Okay, good afternoon, everyone. I think we'll jump straight into it because we took a little extra time during the morning session, which was good because it was very interactive. But we have a lot of ground to cover. But I mean, that's not to dissuade you from asking questions. So please feel free. We'll get into more technical stuff also this session. So then you might have questions. I'll be happy to, answer them and provide any clarifications. Okay, so now one of the things, if you remember, that what we want to study and the parameters that we were talking about was species distribution, right? So we are going to now talk about how do we go about studying species distribution, and I will introduce you to a particular sampling and modeling framework, which is increasingly considered as the gold standard to look at species distribution, primarily because it accounts for certain issues which we'll be talking about. So what we are now, if you recall from what we talked about with species distribution and actually for species richness as well, we were talking about a process of colonization and extinction, right? So we were saying how if green indicates that all of these patches are occupied by any given species, and then when it becomes white, basically it's gone locally extinct. And this can change in time, right? So some empty patches can get colonized, patches that were occupied get empty because animals have moved out. So we are essentially looking at trying to understand all of these different things. And a key thing here, we've already talked about this, we've talked about the fact that even if we go to a particular place, it's not that we will see everything that is there. We can't observe the system perfectly. So there are going to be animals, different species, individuals of a particular species, whatever it is, that we will be missing. And so that's why detection probability, which is essentially to do with the idea that you can't see your system perfectly, so you can't detect your species perfectly, is a very important aspect even when we're looking at species distribution. So when we're looking at species distribution across patches, like I showed in the previous slides, P, small p here, is the probability of detecting a species within a patch given that it is present. Obviously, if the species is not there, you can't detect it. But if it's there, there are two possibilities. Either you'll detect it or you'll not detect it, right? So now when you, okay, I'm jumping ahead of myself. You can also think of it as a proportion or the proportion of sites where the species is there. So occupied sites where the species is detected. So of the sites that are occupied, what proportion of them do you detect the species in? So you can think of it as a probability, you can think of it as a proportion. We'll be talking more about this as we move forward. Okay, so we use a modeling approach called occupancy models or occupancy estimation to get at species distribution. The basic question we're trying to ask is, what proportion of an area, any area of interest, if this is your study area, whatever that is, what proportion of that area is occupied by the species? So what proportion of that entire whatever area will have the species that's occupying that area? Okay. Further questions may be, how does this proportion change over time? So if you're tracking it over time, you may say, how is that changing over time? We may say, what determines which sites are more likely to be occupied? Now we're getting into the why questions. We're looking into what all those different things we said, drivers of change, all the different factors that can influence the underlying vital rates. That's what we are interested in over here. And at what rate are empty sites colonized and at what rate do local extinctions occur? This is all when you're tracking it in time, right? Yeah, fine. So occupancy, if I have to talk about a definition, is the act or state of being present within a particular defined area of interest. Let's say it is a particular naturally defined patch, a naturally defined wetland, or it may be that we've imposed a grid network over our study area. Then it is the act of or state of being present within a given grid cell in that network. That is what we're looking at. Or of it being occupying a particular wetland or particular water body, particular patch. Whatever we have defined as our sampling unit, it is the act or state of being present in that unit. All right? You can look at it as the proportion or the proportion of if you have a grid of, let's say, 100 cells, what proportion of those 100 cells are occupied by the species is how you can look at it. Or you can think of it as a probability. If I take any one cell from that grid network, what is the probability of the species occupying that cell, right? So you can look at, effectively it's the same thing, you can just call it a proportion, and you say what proportion of the total area is occupied, or you can take any one cell and say what is the probability of the species occupying that particular cell. Okay, let's think about this. So this is your entire area of interest, all right? This is your study area. Now we want to systematically sample this whole area to look for any one species, let's say, right? So how do we do it? One way to do it is to put a grid network on it, all right? So let's say this is now our grid network, and the total number of sites we have is 210, where site is each of these grid cells, each of these squares. Now, let's say the number of occupied sites is 70, right, which is indicated by the blue. So occupancy then is 70 divided by 210 is 0.33. So you can say 0.33 or let's say 33% of all of the sites, of the 210 sites, is occupied by the species. Or you can say that the probability of the species occupying any one of these grid cells is 0.33. Yeah, everybody is clear on this so far? Okay, all right. So if we had 21 sites that were occupied, then it would just be 21 divided by 210, that's 0.1. So probability is 0.1 now instead of 0.33, right? Now, if it was all sites were occupied, then obviously your probability is 1. Now, what is missing in this so far? What is missing in this? What are we? What? Say that again. Whatever occupied. There's a number of. Okay, so whatever sites that are occupied, where we have detected the species, right? Those we know that are occupied because we have ourselves seen the species. We have recorded it in some way. We have directly seen it. We have taken a photograph. We have got its signs, whatever it is. We have in some way directly recorded it. What about all these other green sites where we are saying species is absent? Is species absent in every single of these sites? No. Why? It might not have been seen, exactly. Maybe some sites had the species, but we just didn't detect that species, right? So let's come back to that first example. Suppose true occupancy was 70 out of 210, that's 0.33, all right? But the number of sites in which we detected the species, okay, indicated by the lighter blue, is 42. Now, if we didn't account for the fact that we might be missing some animals, or we might be missing the species in some sites, then what would we say is the occupancy? That's why it's called naive, because we are being naive here. So the naive occupancy would have been 42 divided by 210 is 0.2. So we would have said that the proportion of sites that are occupied in this study area is 0.2 or 20%, or the probability of any one site being occupied is 0.2. But what is the reality? That is 0.33. So we have underestimated the probability of any given site being occupied because we didn't account for the fact that there can be imperfect detection. Yeah.
But not detected. How is it quantified?
Yeah, we are getting to that.
Cool.
Yeah, this whole approach is to how do you do that? Yeah, yeah, okay. All right. Okay, so state variables, we've been talking about state variables. So our state variable here is that psi that you had seen earlier. This is to do with distribution. We call it occupancy. It's a proportion of sites occupied by the species. We've already talked about it. It's also a probability that a given site is occupied by the species, right? Now we have this additional nuisance variable. Nuisance variable that we call detection probability. Why is it a nuisance variable? It's a nuisance variable because we are not really interested in quantifying this parameter, right? We are interested in the ecological process. Where are the species found? Why are they found in some places and not found in other places? That's what we are interested in. But to be able to answer that ecological question, we have to also get our hands dirty with the observational process. Like what Divya said earlier, this detection probability is to do with your observation. There's a certain set of sites, some of them you've seen the species, some other sites you've not seen the species. There are two possibilities there. One, that the species is actually not there, or two, that the species was there but was missed, right? So we have to quantify this detection probability, which is the probability of detecting the species in a site given that it is present. So also can be viewed as a proportion. All right, and then the vital rates also we've already talked about. So this is effectively what an occupancy model deals with in its different forms, which also we'll talk about. But let's look at this. You are asking, how do you go about accounting for the fact that we're missing, that there's a chance that we might be missing some animals. So let's say we have our sites here, okay? I'm showing you 7 sites. What do we do? To be able to somewhere, say with confidence whether a particular site is occupied or not occupied, and what Divya had earlier said about even if you want to make any other kinds of inferences, typically you want some replication. You want more than one sample from a given site or given whatever area of interest. So you have replicates, which would mean that each of those grid cells that you saw in that network, you visit that grid cell on 5 occasions. You go there the first time, you say, okay, in site 1, did I see the species or no? Say no. Okay, so you record 0. Go back the next time. Did I see the species? Yes, 1. So if I have a 1 now, what would you say? What is actually the status of that particular cell? Is it occupied? It is occupied. Because we've got at least 1, that means the species is there. After that, we missed it again twice, and then we got it after that. All right? So if I were, for those who are mathematically inclined, is not very difficult maths, but if I were to say, just for this site 1, missed, got, missed, got. Okay. What is the detection probability for that species in that site? 2 divided by, right? As simple as that. So for each site, you have that. This one is 0 throughout, so 0 divided by 5. This one is 4 divided by 5, and so on. All right? So now, this is called a detection history. Okay? So we have a detection history for each of our sites. And we want to estimate a detection probability for our entire area of interest. So we will have to somehow use this information. So this 2 divided by 5, which is how much? Yeah. Okay. So 0.4. So we've got a 0.4 detection probability over for here. 4 divided by 5 is 0.8. So what would you say is the detection probability if you just use those two sites? You take an average of that maybe. Will you? No. Why not? We shouldn't. Why not? Okay. All right. Okay, just park your thoughts, but we have to somehow use this information that we're getting for each of our sites to be able to say what's going on for the entire system of interest. Okay, so but these are the data you are collecting. You're going to every one of these sites multiple times. Each time you're recording either present or absent. And that absent can be two things. It can either be that it's not there actually, or that it can be there but it was missed, and we will be able to quantify it based on what we just talked about. Also, if we did a mark recapture study, which maybe some of you are familiar with because you brought it up at least a couple of times, there also we are basically saying, did we capture the animal, did we not capture the animal? So we are multiple times sampling the population, and we are saying, is that individual that we've identified based on AI or based on some tags or whatever it is, have we captured it or not, right? So that means what? Each of these over here, the individual has to be in our detection history because we are looking at the fate of captures of that individual. But in this case, because it's a site, you can have a completely all 0 detection history also, which is not possible for animals. Anyway, that's just a side point, but yeah.
But pseudo replication.
Yeah.
Are we doing pseudo replication here?
Not at all.
No.
Okay. So if I did, okay. So how do I explain this to you? See here, pseudo replication would be when you're trying to, let me give an example of pseudo, like if you are, what's this?
Consider each of your, you've gone to site 4, right? And say you've gone to those five different places, 5 times you've done the survey. Suppose you count each of those 5 times you've done the survey as 5 different locations that you've gone. That would be pseudo-replication. Over here, we're clearly saying we're going 5 times to that same site number 4. One site, one.
Does that clear it? Yeah. A typical example of pseudo replication that's used is if you're measuring the height of males or females in a population, and then if you were to measure 3 different males, like the heights of 3 different males, and then say, okay, this is the average height, the mean height, then that's fine. But if you take the same individual 3 different times and say that these are actually different people, then you are pseudo replicating, because actually you're measuring the height of the same animal, same person. Here, it's like what she said is the reverse way. If you, okay, she's already explained it.
Same household, like you're measuring the father and the son, it's highly correlated. So is that, do you count that as a pseudo replicate or not?
What are you measuring?
The height in that, in your example of measuring the height.
Okay, I would say that if you went and measured, see, it can be correlated, but I've seen so many short fathers and very tall sons and whatever, vice
But
versa.
we have a scientific basis for
Sure.
correlation.
So what you might do is that you might collect those data. You may not, you may say that, oh, I'll only get one male from a household. Otherwise, you may use those data, but you may say that I will use as a covariate a thing of the family ID also, so that there's some relation that it's okay, these are relatives, might influence the height measurement that I've got, or something like that you might do.
Some sort of a correction factor that you are doing, or?
It's, yeah, it's like I don't know if are you aware of like random effects and fixed effects? And
Yeah, we covered that.
you covered that yesterday, no?
Yeah.
So it's getting into all of that space, but I won't get into that now because it'll take me time and I'll have to finish my occupancy stuff. But yeah, but just this much is enough for this discussion. So, okay, so basically what we're trying to do is that we have a certain biological reality, right, which is that these black cells are the ones that are occupied and the white ones that are not occupied. And then we have, like Divya said, hierarchical. It's a hierarchical model. This is the biological reality. This is what we're interested in, but we can only make observations from that system. So that above is our observational process. And so you can have a scenario where, let's say, this is occupied, but you may have observations of 0, 1, 0. This is also occupied, but you may have observations of 0, 0, 0, right? Or you may have this occupied and you have 1, 1, 1. So the observation process may vary from the biological reality, and we try and use that to estimate our parameters of interest. Did you have a question? Okay. All right. So now, how do we use the information for each of our sites, our detection history, to get at for the what we're trying to estimate for our entire study system? All right. So we've got basically you've got your detection histories, right? And we want to somehow be able to work with all the detection histories across sites.
In this previous slide, so you have done the field observation and biological reality. So there won't be correlated with that because there is a difference reality in biological reality and field observation. Is there any error nullifying methods where you can easily quantify the exact data, where you can nullify the errors? Because that is the error, right?
That is an error.
Error, right?
That is the error. The error is being nullified by quantifying the error rate through the parameter called detection probability.
Okay.
Yeah, because the detection probability is telling you that you are not seeing, if detection probability is 1, that means it's saying that you are seeing your biological reality with 100% accuracy.
Okay.
Okay. If your detection probability is less than 1, that means if it's 0.8, that means 80% of the times you're right.
Can you give an
20% of the time you're
example
wrong.
with any example like that?
You will see it as the presentation goes on.
Okay, thank you.
Yeah? Yeah. Okay. All right, so first thing we can do is we can take a verbal description of the observations that we have seen. Okay, and then what we'll do is we'll take that verbal description and then translate it into a mathematical form. All right, so let's say we have a detection history of 101 for a site. Verbally, what would you say this represents? Say? The site is occupied, okay, and then? No, that is after you've estimated it, you're saying it's a high probability. Can you describe the 101? It was seen 2 out of 3 times. The site is occupied. The species was seen on the first occasion, missed on the second occasion, seen on the third occasion. Yeah, makes sense? All right. Species present at the unit was detected in the first and third survey, not detected in the second survey. Now, if we are to put it in a mathematical form, all right? This is our detection history. The probability of this detection history is that it is occupied, the site is occupied with a probability psi, right? Because we said psi represents the occupancy probability. So it's occupied with the probability psi. It's occupied and detected or not detected, and in probability theory is multiplication. So psi into, it was detected on the first occasion with a probability P1. It was missed on the second occasion, so it's 1 minus P2. If it was detected in the second occasion, what would it be? 2 minus P2. If it was detected in the second occasion, what would it be? P2. Yeah? So P1 into 1 minus P2, detected in the third occasion, P3. Easy? Yeah? Easy peasy. All right. Okay. What about this? Yeah. So there are different models that if you consider them to be different, then you would have P1, P2, P3. If you consider to be the same, you can just say P.
When do you consider them to be different?
You can have one model which says is different, one model which says is same. You can estimate and see, you can look at those models, which we are going to get into, how do you compare models, and you can see which one has more support. One model might say your one hypothesis might be that detection probability doesn't change with anything. Another one might say that detection probability changes with, I don't know, the day on which you're sampling. One day was a sunny day, one day was a wet day, or whatever, rainy day, and maybe that has changed your detection probability. Right? So you can have 2 hypotheses potentially. But if it's different, it would be P1, P2, P3. If it's the same, since they're all the same, it'll be P. Okay, so for this, what verbal description first, please? Not detected. Okay, and what about the psi part? Is not occupied. Yeah, so then what is the other possibility? May be present. So it may be present, or maybe it's not present. If it is present, it was not detected on any of the occasions. Okay, all right. Species is present at the unit but was not detected in any survey, or species is absent at that unit. Or means what will happen now to the mathematical notation? Addition, right? So now we're saying that, let's do this first. Species is not present in this unit. So if it was present with a probability psi, so not present is 1 minus psi. Yeah? Or that it was present, okay, but it was missed on each of the 3 occasions. So 1 minus P1, into 1 minus P2, into 1 minus P3. That's a psi into all of those. Yeah, this is summation for, because it's P1, P2, P3. J is going from 1 to 3. This is multiplication, no, this is not summation. You're multiplying, so you say multiplying psi into 1 minus P1 into 1 minus P2 into 1 minus P3 plus 1 minus psi. Yeah, everybody is with me? So I didn't. Psi, I've been talking since the morning.
No, 1 minus PJ.
Somebody's been sleeping, huh? J is your sampling occasion, which is going from 1 to 3.
I'm saying why we have 1 minus PJ in the first one.
Oh, because we didn't detect it, no, even once.
So, but we are taking in the second, like plus 1 minus psi also, that time we are also taking.
No. There are 2 possibilities here. Possibility number 1, the species is actually present in the site, so it occupies the site. Occupies it with a probability psi. But it is missed on occasion 1. So if it was detected on occasion 1, it would have been P1. So since it's missed, it's 1 minus P1. It's missed on occasion 2 also. So into 1 minus P2. Is missed on occasion 3 also, is into 1 minus P3. That's one possibility. Or that is not present at all. So it's 1 minus psi. Yeah? Okay. So that's how you deal with it. Okay, so here you can have, let's say, 2 possibilities for anything. You can have it's present or it's absent. If it's present, it's with probability psi. If it's absent, it's 1 minus psi. And then you can have it detected. So P1, P2, P3, that becomes your disease detection history. Correct? Everybody follows that line? Let's do one more. It's present, but it's missed on all. So 1 minus P1, 1 minus P2, 1 minus P3. That gives you 0, 0, 0, where it's present but missed here. Yeah, so same way, like basically for all the different detection histories, you can basically start by describing it verbally. It's always easy to describe it verbally first, but after that you can very easily represent it mathematically, and you can get the probabilities for each of the sites or each of the sampling units, each of your grid cells, based on just the string of zeros and ones that you've seen across your replicates. This is the fundamental idea of how an occupancy model works. Yeah? Okay, this is just more examples of how you can go and have different outcomes. We've already talked about that. Okay, now we want to, based on all of these different detection histories which we have now mathematically represented, we want to be able to estimate one detection probability, one occupancy probability for all of our sampling units. Okay, how do we do that? Okay, so we use what is called a maximum likelihood approach. So we basically are maximizing the likelihood of our model given the data that we have. Okay, so how do we do this? So we're saying the likelihood of our model, what is our model here, is our, I can use this actually, our parameter estimates. So likelihood of psi and p given our data. Data is H because H represents histories, detection histories. So likelihood of psi and p given H is equal to the probability of that particular detection history given an estimate of psi and given an estimate of p. All right, and this is done through also you have a particular probability for history number 1, that's site number 1. You have one for history number 2. We have history number 3. You're basically multiplying this for across all histories. And what the maximum likelihood estimation process does, very simply put, is that what it's saying is that there is a particular estimate of psi and a particular estimate of p, okay, that is going to give you the highest chance of getting those histories that you've seen. All right. So it essentially, you can think of it as a graph. On your, let's say on your y-axis, you have psi. On your x-axis, you have p, detection probability, okay? So the potential space in which anywhere you can have a particular estimate of psi or p, which will give you the highest chance of these data, can go from 0 to 1 for along the x-axis for p, and can go from 0 to 1 along the y-axis for psi, okay? So any possibility in that entire space can give you that. So what the maximum likelihood estimator does is that it roams this entire space. It roams, this is my x-axis, this is my y-axis, roaming around over here, and is looking for what is the likelihood of these data given this particular p and this particular psi value. And it again goes somewhere else and looks for those. So it's looking for it, and you basically have like peaks everywhere. You have peaks everywhere, and it's looking for the tallest peak. So where you have the tallest peak is where it will say that for this estimate or this value of psi and this value of p, you will have the highest chance, the highest likelihood of observing the data that you have observed. Okay, so that's basically what the maximum likelihood estimator does. All right. Okay, I hope everybody is clear on that. Yeah. So you will have many peaks. Like for any, let's say psi is 0.2 and p is 0.5, there'll be some likelihood for these data for that. When psi becomes 0.4 and p becomes 0.6, there'll be some other likelihood for that. Each of those represents a peak, but there's going to be one peak which is higher than every other peak, and that is the peak that will basically coincide with that estimate of psi and that estimate of p that has the highest likelihood of coinciding with your observed data. All right. Okay, so then this is how you go about estimating your parameters of interest. Now you want to not just say, okay, what is the probability of a particular unit being occupied by a species, or what is the detection probability here? You want to know why is the probability this much, or why is only this much proportion of the area occupied? Why is that I'm seeing less than like I'm only seeing 20% of my animals? Why? So you want to ask those why questions, right? So many things can be influencing your detection probability. It could maybe to have something to do with the type of habitat, like the forest cover, maybe. It could maybe have something to do with the weather conditions when you were sampling. Maybe how rugged the area is. Maybe you're not able to look for animals well when it was like very steep slopes. Maybe accessibility to areas also had an influence. So all of these things can influence your detection probability. And when you're trying to incorporate these covariate effects, essentially what you're trying to do is that you're trying to say that there is no unmodeled heterogeneity in your p and psi parameters. Like, so whatever variation you see in these, it is being accounted for using some covariates. What I talked about so far was with that one psi and one p was that I'm saying that psi doesn't change with anything, p doesn't change with anything. So there's one particular psi and one particular p that gave me the maximum likelihood of these data, right? But what if psi changes with distance to forest? Further you are from the forest, less chance that the species will occupy a particular area. Could be, right? Many species like to be in forests. They don't like to venture out so much. What if p is determined by something else? So then it's not a single estimate of psi anymore. It's like as your distance increases, your psi will go down, correct? So then we have to account for that heterogeneity in psi that we are seeing in space. And we are using these covariates to do that. If you remember from Divya's presentation, so our parameters are our psi and p. We will effectively be constructing a model, and on our right side will be our covariates, are basically our predictors of why we get a certain kind of psi and why we get a certain type of p. All right. So like I said, this is to ask various ecological questions, to do with what determines psi, species-habitat relationships, responses to human pressures, interspecies interactions. You could be interested in what influences P, because if you don't account for it, then it can lead to biased estimation, and it can lead to misleading inferences if P and psi vary with the same covariate, if you don't account for detection probability. So these are all various reasons for why you might want to go about doing this. Okay. So now, when you're thinking about covariates, there can be two different types of covariates. One is site-specific. So that means it's to do with, if you remember our grid network earlier, each of our grid cells there will have a particular covariate value. Let's say percentage forest cover. Percentage forest cover that changes from one grid cell to the next, but for that grid cell is the same. So one grid cell has 20% forest cover, one grid cell has 80% forest cover, one grid cell has 40% forest cover, and so on. So you're trying to say, oh, does my percentage forest cover have an influence on my occupancy probability or not? All right. Then there can be others, like so habitat type, patch size, canopy cover, etc. Then there are others that are to do with the survey itself. Survey itself meaning the replicates. We had 5 replicates. So what if something changes from one replicate to another? What will that tell us? That will tell us something about detection probability usually, is like weather conditions, who's observing on a particular day. Today, suppose I went and I'm useless at looking for like whatever, some amphibian, but somebody else goes, is like a great, herper, and he just goes finding animals everywhere. So his detection probability is going to be much better than mine, right? So those are survey specific. So that's changing from one replicate to another. If it changes from one replicate to another, then to come to your question, it'll be P1 will be different from P2, will be different from P3, because in each of the replicates, there's a different P. And that's related to maybe the observer, is related to the weather or something else. Okay? All right. So, very simply, when we are talking about the parameter and the covariates, or the response variable and the predictors, what are we looking at? What does this represent? Huh? Straight line, no? It's a straight line, correct? This is your y, this is your x, this is what? Intercept. And this is your slope. Everybody knows then what slope and intercept represent. What's the intercept? It's where the line, what does it do? It intersects with the y-axis. What's the slope? The rate at which it's changing, right? So now, if we're talking about a straight line, if it starts at 0, suppose it's going up, suppose the slope is positive. Theoretically, how far can it go? Infinite, right? What about your occupancy parameter? What can it go? What is it bounded by? How far can it go? It goes from 0 to 1, right? It can't go beyond 1. Detection probability also can't go beyond 0 or beyond 1. So these are bounded by 0 to 1. So this is for a normally distributed kind of response that you're looking at, but ours are not. Ours are like 0 to 1, right? So then what do? We have to somehow deal with the fact that we want a straight line, but we are not dealing with a response variable or data that are amenable to looking at it as a straight line. So we have to somehow transform our parameters such that it starts looking like a straight line. So what we do is what is called a logit transformation, or we use a logit link. So psi is not equal to a plus bx, because psi goes from 0 to 1, so it doesn't represent a straight line. But logit psi represents a plus bx. Okay, and what is logit psi? Logit psi is the same as log psi divided by 1 minus psi. So log of psi divided by 1 minus psi is the intercept plus slope into the covariate that we are interested in. Does everybody follow me? Everybody is familiar with straight lines, right? So what we are trying to do essentially is this is our covariate. This is our covariate that's going to influence how our parameter of interest, which is psi, is changing. Let's say this is distance to forest, this is percentage forest cover, something like that, okay? And we are saying that there are different values of this and there'll be a slope associated with this, there's an intercept, and this is, to make it a straight line, we've used a logit function, so therefore it becomes log psi divided by 1 minus psi, all right? Only when you have this, can you look at this as a straight line. And then when you want to actually estimate psi based on that, you have to what is called back transform. How do you back transform? You want to get rid of this log. How do you get rid of a log? You do exponent. So if you do exponential of this, then this becomes all exponent, right? So if you solve this, you'll get e to the power of this straight line divided by 1 plus e to the power of that. Okay, so this will give you the real estimate of psi. But this will let you look at the relationship between a particular covariate and the slope associated with it. Positive relationship, negative relationship, all of that. Yeah.
Just to be clear, we are not actually, we are actually considering the probability of getting ones and zeros in that.
Yes.
Logic, not 1 or 0. We don't just put 1 or 0 in that.
Yes, yes, it is a probability.
It is the probability of getting 1 and the probability of getting 0 that.
It's the odds ratio of the ones and the zero. Yeah. Okay. So because we were able to do that transformation, we can now model these coefficients, like I said, like as a function of these different, like the x's that I talked about, the covariates that we are there. And then I already said inverse logit link or the reverse transformation will let us get to the, or back transformation will let us get to our actual estimate. So suppose we have altitude as our covariate, okay, it's on our x-axis, goes from low to high, and our relationship was that it was a negative relationship with altitude. If we didn't do this back transform, so this is going from 0 to 1, that's why it curves down like this. If we were looking at it at that other scale, then this would be a straight line. Okay, but we've transformed it such that we can get at the estimate of psi going from 0 to 1. So at very low altitude, it's 1. As the altitude increases, this estimate goes down. Yeah? Okay. All right. So few things to just keep in mind. So your intercept, which was your beta naught, right? It controls the position of the transition from a 0 transitioning to 1. Okay? So if you have beta naught of 3, which is here, beta naught of 0 and beta naught of minus 3, you can see that the position of where it's the same slope. So it's showing the exact same shape. The curve is showing the same shape. But the location where it's transitioning from 0 to 1 is changing based on what the intercept value is. All right? The sign of your slope will tell you whether it's a positive or negative relationship very clearly, right? If it's negative, it will go down. If it's positive, it will go up. Then this is what we were talking about. What is the slope earlier we said? And you said it's like the rate at which like the thing changes. So basically it's the rapidness of the transition. This is a slope with a value of 0.4, right? This is slope with a value of 1 and this is slope with a value of 1.5. So clearly, like you can see the change at which is going from a gradual S to a sharper edge, S, sorry. Okay, and if you have like really like large slope like this one, which is V1 is 20 here, this is the red line, it shows a very abrupt transition. It's like 0, 0, 0, 0, 0, 90 degree growing up and then 1, 1, 1, 1. Yeah, so because you are going from a gradual, like you're going gradually to like that. It's all changing with your slope estimate. Okay, so same way as how we did we use the logit function for our psi parameter. The detection probability P also is the same thing, it goes from 0 to 1. So again we have to use logit P is equal to beta naught plus beta 1 into x whatever, the different values of x. So again you have to use back transform and get to your P estimate. Yeah, so essentially, since yesterday you guys talked about GLMs, did you talk about logistic regression? 0-1 data. So essentially this is the occupancy model is also a logistic regression. You're using data that are 0-1 data, presence absence data, but actually not presence absence. Why? Because it's actually detection non-detection data, because you also have the detection process. So you effectively have two logistic regressions slapped on top of the other. One is to do with your occupancy parameter psi, the other is to do with the detection parameter, that is P. All right, and they're most powerful when P is less than 1. If P is equal to 1, it's like a logistic regression. You can basically throw out the P and just look at psi. Right, you can ignore imperfect detection, and if you have a good data set, you have good study design, then you can definitely tease apart the confounding effects of P and psi. So it's a really powerful tool. It's really for species distribution models. I think this is, like I said earlier, the gold standard, because you're accounting for these biases that can creep into other studies that don't look at imperfect detection. Yeah. Confounding effects, because suppose your occupancy parameter, occupancy of tigers was influenced by let's say the prey density, and your prey density was also influenced by or your observation of your tiger presence was also influenced by no, this is not a good example. Okay, let me think of an example, but basically what I'm trying to say is, if you have two different I had talked about earlier about how rugged a particular area is, how up and down an area is. Suppose for a particular species, the up and down of the area is actually also determining where it wants to be or not want to be. Okay, so occupancy is impacted by that. So the species doesn't like these really steep slopes. So it likes like nice flat areas. So it actually occupies the flat areas more. Okay, but your detection probability, let's say, is also influenced by your the ruggedness terrain. Actually, let me rephrase that. The species likes, it's a slope loving animal. Let's say it's a mountain ungulate, mountain goat. It likes the slopes. Okay, so the ruggedness influences the occupancy parameter, but as you have more ruggedness, you have higher probability of occupancy. Okay, but we are also sampling this area, and we humans are not able to go scaling up those areas. So we tend to like miss this animal a lot more on the steep slopes. So if we didn't account for the detection process, we would say that because we've not gone or not been able to go to the steep slope areas, we would say that the species is found more in the low lying areas. Okay, but actually that's not the case. Actually they are found on the slopes. It's just that the detection on the slopes is lower than the detection below. So there is a confounding effect here of what is the ecological process and what is the observational process, and we need to be able to separate those two things out. That's what is meant by the confounding effect. Sorry, I couldn't come up with an example well, but hopefully that clarified that. Yeah. Huh. Yeah, same thing. Canopy dwelling birds, they'll be on top of the canopy, but our detection is going to be lower there. So if you don't account for the detection probability, then you will interpret those data in a biased way. Yeah. Yeah. That's what I was trying to go with that tiger and prey example, and I kind of couldn't. couldn't put it, that's again a confounding effect. So if you have not occupancy, but abundance, if you have a higher abundance of a particular species, then maybe your probability of detecting it is also going to be higher. There'll be more signs left behind, right? Huh. Huh. No, 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 because your occupancy is your parameter, right? So your occupancy is a response and it's coming from, and you're quantifying it based on your 0-1 data, right? So you cannot use it as a predictor or a covariate, but you might have something else that could represent, I don't know, like so density of the animal, for example, like, okay, coming back to the tiger and prey example, let's say it's the prey species. Maybe you had prey density as a covariate. You had prey density as a covariate, or you had livestock density as a covariate, or the encounter rate of livestock as you did your surveys as covariates. You could use that because you're saying that where there is higher prey or higher livestock, the tigers are using those areas more. Okay, but this covariate is representing that influence. So you're not using the occupancy, you can't, because that's your response, but you can use covariates that answer those kind of questions. So you have to think, like Divya said earlier, you have to think a priori about what your hypotheses are about your system, what's influencing the distribution of a particular species, and then you have to answer the questions accordingly. Yeah? Okay, so I won't spend too much time here, but I just want to say that once you've got through the maximum likelihood approach, you've got like an estimate for psi and p for a particular model, okay? And you have like 3-4 different models. In one of them, you have forest cover as a covariate for psi. In another one, you have, I don't know, like thing. You can have like, what else did I say? Village density as a thing of psi, distance to forest as another covariate for psi. All of these are different models. Like Divya said, you can have additive effects of them, or you could just have them individually, or you could have an interactive effect. These are all different models, and now you want to compare these models to say what explains your data best, right? So the way to explain or look at the models is to use what is called, so this is within the model selection within an information theoretic framework, okay? It's called information theoretic approach. So here you're using Akaiki's information criterion, which is referred to as AIC. So what AIC basically does is that remember we talked about the likelihood given your data previously, likelihood of your model given the data. So that's basically it. It's -2 log the likelihood of your parameter given your data, and you are penalizing this for the number of things you have, parameters you have in the model. Okay, so for example, here parameter is not just your psi, but is also your different covariates that you might use. So something which is a simple model would just be psi is a function of forest cover. A more complex model would be psi is equal to is a function of a forest cover plus livestock density plus village density. So the second model is more complex, and maybe it explains your data better, but what AIC does is that it penalizes it. It has to explain your data better, like more better than when it is just using the simple model. So there's a penalty term associated with this, and how you try and compare models is to look at the AIC scores and lower the AIC score the higher support that particular model has. And that's why you have this penalty term associated with the number of parameters that are being used in the model. So it's basically a trade-off between fit, how well the model is fitting your data, and the complexity that you can build into it. So you want to have a sufficiently complex model that gives you nice understanding of your system, but not so complex that you're just throwing everything into that model and seeing if it's the best model. Because as you're doing that, maybe it's not giving you more information than a simpler model might be giving, and that will get penalized because of this plus 2k over here. All right, so basically what you do is you have let's say one model and you have an AIC score for that. You have a second model and you have an AIC score for that. You have a third model and you have your AIC score for that. You compare the AIC scores for each of the models, and the model with the lowest AIC score has the highest support. And the general rule of thumb is that models that are within, so the model with the lowest AIC score, you can then subtract the AIC for the other models from that model, and that's like called delta AIC. So this is the AIC for a particular model minus the minimum AIC. All right, so you're seeing the difference between the lowest AIC and the AIC of any model. So the model which has the lowest AIC, the delta AIC will be what? 0. And then as the AIC scores increase, there'll be a positive amount, let's say 1 plus 2 plus 3 and so on. So the rule of thumb is that models that are within delta AIC of 2, so anywhere going from 0 is the best model, and up to 2, you're saying that those models are comparable in terms of their support. And if that's the case, you may want to make inferences not from just one model, but more than one model that has a comparable score. Once you go from 2 to 7, the value is like, the support becomes less and less, and they don't have that much support. And beyond 7, you can actually disregard the model. Beyond 10, actually, you're saying that that model has doesn't explain any of the variation in your data. Yeah.
Minimum value of that AIC minimum, should that be in a particular range to consider it as a good value?
Oh, no. So your actual AIC value doesn't matter at all. Your actual AIC value, doesn't matter at all, but it doesn't matter because you're not interested in is your AIC value minus 24325.7 versus like it's the versus another model which is minus 23125.8. It's the relative, the difference between the two is what you're interested in, not the actual AIC score that you get. The actual AIC score is your minus 2 log likelihood of your data given your parameter given your data plus the 2 into the number of parameters that are there in the model. That's your actual score you'll get. But that score is not really of interest. What is of interest is how does that score compare to all other models and their scores. Yeah, and you want to have minimum AIC, which will be your top model, which will be 0 AIC, delta AIC, and then delta AIC increases. And all those delta AICs that are within 2 are actually comparable models, and you should make inferences from them. Maybe they represent different hypotheses, and you can talk about, oh, as per this hypothesis, this is the effect, and as per that hypothesis, this is the effect. Yeah, but that's how you basically go about doing model selection. You use your AIC values to look at your scores. Yeah, I don't think we should get into too much of this now because we don't have time. Yeah, so like I said earlier, think about your models before, see if they're biologically plausible. Like it shouldn't be like some nonsensical model. There should be some ecological expectation for why a particular covariate influences your parameter. Check for convergence. Well, convergence is when sometimes you'll have a model where you'll get like really absurdly large estimates of your slope estimate will be like minus 20,000 something or whatever. Then those doesn't make sense. The model has not converged. And what do I mean by not converged? It means when it's looking in that MLE space, in the maximum likelihood space, for those peaks, it's not found that highest peak. Okay, it's not found the highest peak. Maybe your sample sizes was low or some other reason. It was not able to find that peak, so then that model cannot be can be considered. Maybe you had too many covariates in that model, and for the number of covariates, you just don't have enough data to really be able to make sense of it. So you have to check for convergence. You have to check for multicollinearity. This is very important. That means multicollinearity means that the predictors or your covariates should not be correlated to one another. So you need to have independent covariates that are, so if you have an additive effect of 3 different covariates, each of those covariates should be independent of each other, which means that there should not be a significant correlation between them. So you have to do like a Pearson's correlation test or something before that to look at how correlated they are, and you shouldn't use, let's say, distance to forest and distance to protected area may be correlated because mostly protected areas have forests. So then you cannot use both in the same model. Saying psi is a function of distance to forest plus distance to protected area. That would not make sense. Yeah. And you have to think about these interactive effects also properly. Would it make sense to say that, oh, one thing varies as a function of another depending on the value of the other covariate, like we talked about earlier. If there's high, like a, let's say, give me an example. High canopy cover and low canopy cover are your two different things. Do you see? If I give an example of interactive effect, I'm not being able to think of one.
High disturbance, shrublands has an effect. If there's low disturbance, shrublands
Yeah. Okay, I can give an example, and I'll give an example of elephant study that I did. So you have distance to protected area with the hypothesis that elephants are kind of being restricted to the protected area because of human disturbance outside. So you're saying that as you move away from the protected area, the elephants will be using that area less. Okay, so that's one hypothesis. And related to it, you're saying that why are they not using certain areas? Because of human disturbance. So clearly village density may be a covariate that you're using to say that it has an effect on this elephant occupancy, let's say, or site use. So now, in the absence of villages, you can have potentially elephants using even areas outside protected areas. No, they can move further because the disturbance is what is determining them. So you're saying there's an interactive effect of distance to protected area and village density. Low village density, even at far away distances from the protected area, elephants might be using those areas. High village density, even next to the protected area, you may not have elephants using those areas. So that's then an interactive effect that you thought of a priori, that okay, these are the reasons why we expect an interactive effect, and then you try and actually test for it. Okay, this is what I said. Strong support for a model when delta AS is less than 2. Weak support is between 2 and 7. Greater than 10 is like no support for a thing. You had asked about your question about whether it's a good, that AIC score, is it a good score or a bad score? Since we were talking about relative, all this time we were talking about comparing one model to another and saying which model has the lowest AIC scores, right? So you're basically saying among the set of 10 models that I'm testing, or 5 models that I'm testing, which one has the most support? But what if you've taken 5 really faltu models and then you're comparing among them, right? Yeah, so for that you have what is called a likelihood ratio test, all right? It uses the log likelihoods, the -2 log likelihood of the parameter given the data that was there in the AIC scores, that part. It's also referred to as deviance, so that -2 log likelihood, it uses that and looks at the likelihood ratios to say, is your, forget if you're comparing across models, is a particular model actually fitting your data well or not? So that would be a useful thing to also look at in addition to comparing between or among models. All right. Yeah, I guess. Yeah, look at your entire model set, not just the top few. Convergence is an issue. Yeah, if you don't have, like I said earlier, if you don't have a best model, there's not one model where there are like 2-3 models within delta AIC of 2. Then you can use inference from all the models with support. You can also do model averaging. I won't get into how you go about doing model averaging, but that can be another way of saying that let me get at an average estimate of parameters of interest from all of the models in my candidate model set. Okay. Okay, this I've already said. Yeah, fine. I think that much is enough. Yeah, okay. Yeah, sure.
I think so, based on
GLM and also looking into the analytical aspects. Could you please also kind of touch upon on some things like the sample size, like how do we people decide how many grids and how many
Yeah.
things to select depending? Yeah, and the power analysis, some little bit if you can touch upon, that'll be very useful. Thank you.
Yeah. No,
Because
thank you.
disease data get very skewed and then prevalence and all of that gets
Yes,
affected by that.
I will very quickly you say, thanks for that question. I actually didn't have enough time to get into those aspects, but they are important. So I'll talk about it using this example. Okay, this is an example from Karnataka. This is what is referred to as the Malnad landscape. So Bangalore is there, and this whole area, so there's Nagarhole, Bandipur, people, you guys must have gone, right? So, and then there are other protected areas over here. And this whole area has like potential habitat for number of different wildlife species, including this particular study design that you see. This was for a study on tigers. Okay, looking at tiger occupancy from here all the way till there. All right, this whole area of interest. Now, two things to keep in mind here in terms of sample size. In terms of sample size in general, so one of the main like fathers of the occupancy model is a person who did his postdoc with the actual father of the occupancy model, whose name is Jim Nichols. So Jim Nichols is somebody who's really like thought really hard about these models. But the person who did a postdoc with him and really took this forward was a person called Daryl McKinsey. So Daryl McKinsey has, I mean, they have a book on occupancy models. There are many papers on them. And in their book, as well as in a paper that Daryl McKinsey published in 2005, they've done like a power analysis to look at, for different estimates of psi and different estimates of p, what is the sample size you require? How many grid cells do you need to sample? And how many replicates do you need per grid cell? Okay, so in general, I mean, I would encourage you to look at that if you're interested in doing an occupancy study. But in general, I think the minimum number of sampling units or sites that you require to be able to even run like basic occupancy models is about 40 sampling units or 40 grid cells. Like if you're using grid cell as a sampling unit, you're using a pond, then 40 ponds. Whatever is your unit of interest in which you're making an inference. So more than that, the better. This entire grid network that you see has 210 cells. Okay, so obviously if you're getting close to around 100, then you're well placed to use more complex models if you have more complex questions to ask. That's one thing. Replication, you would require a minimum of 3 or 4 so that you have enough zeros and ones to work with. Ideally, if you're in the 6 to 8 replicates, I guess between 4 to 8 if you've got, I think that's usually enough. For most cases, if you have a really hard to locate species, very difficult to detect, you may need to have more replicates. You just have to, your search effort needs to increase, no? You have to look a little harder. So then you may require a slightly larger number, let's say 10, 12, but more than 12 replicates usually are not required. So these are just general rules of thumb, but I would encourage you to look at Daryl McKinsey's work on that. The last thing I want to say here is about your sampling unit. Now, all this time we've been talking about occupancy, which is the probability of a species occupying a given unit of interest. That unit in this case is a grid cell. For most of the examples, it was a grid cell. Sometimes that unit could be a naturally defined unit. It could be a pond. So let's say talking about the occupancy of some frog species in a network of ponds. So you have a network of ponds and you're saying for each of these ponds, what is the probability of frogs occupying that unit? So the unit is automatically or naturally defined and is naturally closed because this frog, let's say, is not mobile enough to hop on from this particular pond to the next pond within a short period of time, right? So it's effectively closed, and then when you've gone there, if you've got a 0, you can say that either it was present there and was missed, or it was not there, right? What if it was there, the first replicate, and then it hopped out and went away? So then your whole thing gets biased, right? So that's why you want to, when you're talking about occupancy, you are talking about a size of a unit which is large enough to encompass the home range of that particular species. So in this tiger example, each of these grid cells is 188 square kilometers. So it's 188 square kilometers, so that you're making sure that a tiger, even if it's moving around, when you are doing your search, you have it's still a closed system. It's not that the tiger can move out of there permanently. It will move around within the unit. So that's the thing. So usually, if you're trying to look at occupancy, you will use a sampling unit that is naturally defined and will encompass the movement of that animal within that study period, or it will basically be large enough to encompass the home range of the species. Just to say that in case it is a smaller unit, then your occupancy probability becomes you can still use the same model in the same way, but your interpretation of that parameter will no longer be occupancy probability of the species occupying a unit. Instead, you will talk about probability of use. What is the probability of that species using that particular unit? Because it is using multiple units in its home range, but is the probability of using that particular unit. It's basically just different orders of habitat selection. Effectively, you're looking at smaller units there. Only thing you have to keep in mind is that any change in that occupancy status in that smaller unit, if it changes, let's say the animal moves from one unit to another, that change is random and not like directed by something. Let's say if you did it across a season and it changed during the wet season, they all moved away from one area to another area, then that would not be a random change. It would then cause some biases. So those are the main things to keep in mind. Did somebody have any questions? No. Yeah.
You mentioned about the values that are given in the book. So how did those guys arrive? Was it experimentally done, or did they run some simulation?
They ran some simulations.
Simulations, okay.
They used some field data and then they ran some simulations. They basically studied salamanders, and then they took different data samples, and then they did some simulations and they came up with. Okay, yeah, so the tiger example I've already said. So 210 cells, 188 square kilometers each cell, covering this whole area. There are multiple replicates in each cell. They were able to estimate the tiger occupancy across. So it ranges from 0.29 to 0.77 here, like going from brown to green. So you can see how it is varied in space. Similarly, work has been done on dholes, Indian wild dog, same study area, and the darker the color, the higher the probability of dholes occupying a particular unit of interest. Same thing, we were also involved in doing some work on elephants. So just to show that at very large spatial scales, you can look at distribution of species, gives you really good spatial insights if you're talking about it for a particular season. So the occupancy status can change in time, like 5 years down the line, maybe this dark blue will no longer remain dark blue. But at least for the period of time that you have done your survey, it is closed, what is called geographically closed. That means the animal is not moving out of the system during that period of survey. Yeah, but if we say, everybody tired out by all this, or I just have a few more things to say, and then we'll be done.
AIC criteria, right? So I just want to ask, is there any criteria for the parameter selection also? Suppose you have any data, right? So is it possible to select the parameters, that which parameters we have to estimate from this particular data? Like you are saying in the MLE method, you have k number of parameters, right? So like there are more parameters. So how can we select that these number of parameters?
No, so it's a bit confusing because the same term parameter is used both for what you're trying to estimate as well as what you're trying to explain the estimates with. Because it's, okay, so our initial use of the parameters was what we were referring to as the response variables. The response variables in the occupancy model are only two, which is psi and p, occupancy probability and detection probability. That's all you are estimating. Nothing else you can estimate, okay? in the basic occupancy model. Now there are all these different covariates that we use to explain the variation in this. We modeled it. We modeled psi as a function of distance to forest, forest cover, whatever, like other things like that, right? Now, in the AIC framework, when you're trying to look at model selection, the parameter that you're estimating, which is psi, is counted as one parameter, right? And then you have for each of the different covariates that you're using. Suppose there are 3 covariates for that model. So that will be what? It will be intercept beta naught plus beta 1 into x, which is, let's say, your first covariate, x1, which is your first covariate, plus beta 2 into x2, which is your next covariate, and beta 3 into x3, which is your third covariate. Okay. Now, because there are 3 covariates, those are also counted in the model as the number of parameters. So you have psi plus x1 plus x2 plus x3. So that makes it like 4 parameters.
Basically, whatever you're estimating is a parameter.
Yeah, but ma'am,
So either you're estimating, if you're just doing a basic model, either you're just estimating psi and b. Otherwise, suppose you're modeling it based on distance to forest, you're no longer estimating psi.
yeah.
Psi is equal to some beta naught plus beta 1 coefficient into that covariate value. You're estimating now that coefficient.
Yeah, it depends on the model, right? Like which data we are using. But this is not related to the ecology. I'm just asking generally. Suppose you have a particular like 2 data you have. So how can you decide that the parameter is not relevant to the data? So how can you decide? No, if it like, okay, wait.
You have to.
If it is not,
Let us just take a step back. So the data you collect is dependent on the questions you're asking. The models that you choose is dependent on the questions that you're asking. Each model will have a set number of parameters. How to choose the best model comes from model selection, from the model selection approaches. And based on the model selection approaches, you have that number of parameters. But if you are thinking of, you have completely different data set. One where there is not distribution of species or some species, but it's counts. That's a completely different question.
okay.
So it starts from your question.
No, suppose you have not a different model. You have a single, suppose you have a complex model simply. I'm not a one.
So no, I think I'll answer from what I understood what you were asking in the
Like
beginning.
I'm not from a ecology
Sure.
background,
But what I would suggest from what I understood of your question is that how do you decide what covariates to use?
yes,
Right.
to estimate.
So for that you have to have some biological expectation. There's a question that you're trying to ask. You're trying to understand something. Why is this influencing that is a question for you. So if you're saying why is forest cover influencing the distribution of some species, if that is your question, then you have to include that as a covariate. So what covariates to use will be determined by your biological question.
Yeah.
From there.
Okay, I'm from a epidemiological background actually. Okay, so ma'am, like simple SIR or SVIR model we are taking, right? So we are taking the transmission rate and the vaccination rate, right? Suppose if we are using the number of cases, daily number of cases or cumulative cases, so is it possible to estimate vaccination rate from that daily number of cumulative cases? Because it is not possible that you are using the cumulative cases for the transmission rate and you are using vaccination data for the vaccination rate. Like we are using the one data for the model, not the different. Ma'am, it is possible to estimate vaccination rate from the cumulative number of cases of any infectious disease. So how can we take the vaccination rate? So that's my question. Like if you have a SVIR model, right? We have suppose 5 parameters.
I'm going to guess. Basically, maybe your question will be, does vaccination rate affect number of cases, right? So then the way you collect the data so then vaccination rate is a covariate. Is that right? No?
It could be.
Yes, yes.
It is a covariate.
It's a covariate. And then you look at a model where you estimate that covariate. That's it. Yeah. You get data on vaccination, you get data on cases. Yeah. No, don't simply assume some model. This is not, yeah. No, those are not the same kind of models. They are different models. Yeah, they are different models. These are models, this is modeling used in a different sense from that, right? Those are kind of process-based models. Here you're trying to understand how these covariates are impacting, right? Yeah.
For that, you use your biological expectations, some question that you're trying to understand.
That
will
again
be-
it
that will
depends
be
on the
det-
question.
The criterion is your question.
Yeah.
It's what
Whether
question
you
you're trying
think
to
those
ask.
are important for that question. So for example, with say rate of occurrence of diabetes, I don't think vaccination rate is relevant. So you have vaccination rate, you have like who got BCG, who got blah, 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 mumps, whatever, polio, whatever. It's irrelevant to diabetes, right? That way. I guess, I don't know, I hope that's.
Yeah.
So you have to like estimate the covariates which are actually contributing for your response variable. Is that your question, right?
No, you have to think what they are.
Yeah. So in that way, the first one is relevance, whether it actually affecting your response variable or not. And the second one, you should check for multiple collinearity. So with that way, we can estimate that covariate is really affecting your responsible variable or not. So that is called a variance inflation factor. So if you put your covariates with that responsible variable, it will be giving the values 0 to 5. If it is in a positive way, it is really highly correlating with your responsible variable. So in that criteria, you can consider your covariates.
Okay,
that's
thank you.
fine,
I
yeah.
think we should move on because we're like, quickly out of time. Anyway, so we were saying that if you then wanted to extend it to in. So I'll just then not talk about the multi-season. No, I'll just quickly say that if you want to look at it over time, then you just have to also estimate these colonization and extinction parameters in addition to the occupancy parameter. So you will basically look at the same kind of way how you did verbal description, no? Extinct, went extinct, didn't go extinct, did it colonize, did it not go, did it not colonize, and then you have your mathematical notation. We won't get into it. But basically, using that, you can estimate, so your psi, but also your probability of this local extinction, probability of colonization, and then you have your detection probability. So this is referred to as a multi-season occupancy model, because then you can look at it over time and look at the dynamics. And I will not get into all this, but just the last slide, and is I guess relevant to like what we were just talking about also when it comes to disease ecology. Here's an example of where it was applied, occupancy models have been applied in the context of disease ecology. So you have two things here. You have your host, which is wild blue tits, all right, and you have a parasite, which is your malarial parasite, some Plasmodium species. And the interest was to look at, firstly, they did an occupancy survey to look at the distribution of the host, which is of the blue tits. And then after that, to look at the detection of this parasite in the host. And what they found was that the detection rate in the blue tits of the Plasmodium depended on the parasite load that the host had. So higher the parasite load, higher the detection rates were. So just to give you an example of how basically an occupancy modeling framework can be applied to cases where detection may not be perfect even in the case of the disease or in the case of the parasite.
So given kind of harking back to what Farah was saying, mostly in disease ecology, prevalence is very low, right? So we have really, really low rates of prevalence. So do you think that, I mean, why are occupancy frameworks even more appropriate in such situations? Maybe if you can just say a few words about that.
Yeah, absolutely. Thanks, Uma. Yeah, so because the prevalence is low, it's very much like, going to any kind of forested system and where you're looking at species that are by naturally occurring in low densities, and you many times don't find them. Like if you talk about carnivore species that is elusive and rare, you have to put camera traps and you have to look hard to be able to find it in the system. And you need modeling approaches which will let you use that really sparse data that you're getting to make really credible and useful inferences about the system in terms of, let's say, the distribution of that species. The same thing is very easily usable and translatable in the context of disease ecology because of the low prevalence of different kinds of diseases or parasites. You can, because of the low detection rates, you might be missing, in many cases there might be false negatives where you are missing the parasite when it's actually there, or you're missing the pathogen when it's there. And this kind of a modeling framework will let you account for those false negatives. All this time, basically what we were doing with those zeros is that there can be false negatives where the species is present but we missed it. Same way the pathogen can be present but we miss it. So this modeling framework readily lends itself to estimating that probability of missing that pathogen given that it's present in whatever samples that you've collected. So that's why it's I think quite useful.
Also then looking at it in the context of communities.
Yes.
So basically when you have, how about things like interactions or transmission?
That is something Divya is going to talk about through the MSOM. Yeah, so I just to quickly say, so there are, we just talked about a single species occupancy model, but there are, and Divya is going to talk about that, so I won't say much about it, but there are now occupancy models that look at two species systems, multiple species systems, how the presence of one species influences other species. So all of those very easily then can be considered in the disease ecology kind of context.
I think you have. Oops. Okay, I'm just going to very quickly run through this a bit of this presentation and focus mainly on the extension of the occupancy model that Varun was talking about. There are two things really that till now we've been talking about where's the species, but sometimes we want to put that together and look at multiple species together within a community. So if you're looking at birds, we want to look at all of the different bird species together. If you're looking at rodents, we want to look at all of the different rodent species together, and so on and so forth. We then go back to this question of how many species are there, and potentially after that we can go to a relative abundance. So to look at diversity. So if we want to know how many species there are in a system, then all we need to do is go out and count the number of species, right? But what have we been talking about all of this time? You might miss some species. So that is the basic problem with just going out and counting species. This is a problem that's been acknowledged for a while now. And earlier, there were certain ways in which people could account, or people used to account for it. And these ways have been used extremely frequently. So one, and this is something that you might have seen before, is what is called a species accumulation curve or a rarefaction curve. Idea is the more you sample, the more number of species you'll get, and at some point of time, it will level off. Right? So you're looking for this leveling off point. Yeah. So you go out, you sample some area, you double the area, you first get only one species, then you get 3 species, then you get the 4th species, and so on and so forth, till you get a leveling off of the area. So you can, this can either work with area, this can work with number of individuals you sample. As you sample more and more and more individuals, again, you're expecting that there'll be a leveling off. But it's not as fancy and like model driven and so on and so forth. You're not really estimating what are those number of missing species, right? So we could potentially do better. There are also some,
was not working for the recording. Okay, now is it fine? Yeah. Okay. There are also some estimators that people have used, like the cow estimator, which basically takes into account the number of rare species, right? So the more number of rare species you have, the more chances there are that you might miss out a species, right? Generally, if you have rare species that have only detected once, then there might be species that you've never detected at all. So these estimators are based on that. Over here, you can see what we're trying to get at is F0, which is the number of species that we've detected 0 times. And we use the number of species that we've detected only once to kind of inform that number. This is the number of species that we've observed. This is what we are saying is the actual number of species that are out there. The same thing, I mean, there are different estimators, different cow estimators. If you've got multiple samples, but in the interest of time, I'll just go over this. There are also jackknife estimators. This is also very commonly used. Some of you might have used jackknife estimators for various different things, right? Jackknife, bootstrapping, something. Okay, so a jackknife estimator, it's called jackknife because anybody has used a jackknife. It's a rough and ready knife. Like you just open it, you can cut anything and close it and then put it back in your pocket. So a jackknife estimator is like that. It's a rough and ready method, which has been used for a really long time. What it's called is leave one out resampling. So you have 10 samples, you leave one out, see what happens. You leave the next one out, you see what happens, and so on and so forth. So a jackknife estimator is based on that. You essentially leave one sample out, you see what happens, and then you go through sequentially by leaving each of your samples out and use that to get at what happens, like what happens if I get one more sample. So essentially what have I missed by missing something in your data itself, you understand? No, so you have 10, you leave out the first one. Then you bring that first one back, you leave out the second one. Then you bring that back, you leave out the third one, and so on and so forth. So you're saying, okay, what happens if you leave out one of your samples? Then what is the additive, like what is going to happen if you leave out that sample? Using that information, it gets at what have I missed by not adding more samples. No, you can have first order jackknife, you can have second order jackknife as well. So there are basically like now closed equations, so closed form equations for first order and second order jackknife estimators. This is your first order jackknife estimator. Essentially, you're just adding the number of species that you've seen only once. Your second order jackknife estimator, you're using both the species that you've seen only once and the species that you've seen only twice. These are all fine. They work okay, but they don't work fantastically. And they don't do what you've been talking about all of this time, which is that they don't really go out and estimate how many species have I missed. There are a couple of methods that do that, so we want to focus on those. One is what is called capture-recapture. All of you know that the tigers have been estimated in our country using camera traps. Okay. All right, so it's not that they go and photograph all of the tigers. They photograph some of the tigers and using that they identify what is the number of missed tigers, right? The same approach is used for species as well. I've gone to a location, I have sampled 10 times, I have counted 20 species. How many species have I missed? Yeah. So it actually estimates how many species have I missed, and it can take into account what is called the MH or the heterogeneity detection model, which is to say that each species will have a different chance of being missed. Because there are some species, like if you take birds, there are some species like hornbills, which you may see more. There are some species that are really quiet, you might see it less. Some species are really rare, you might see less, and so on and so forth. So there are the visible species and the non-visible species. It's able to take that into account. But that, basically you take an entire area, you say what is the species richness in this entire area, and you estimate the number of species that you've missed, and you've got a species richness of the entire area. It doesn't go to the level of detail that Varun was talking about till now for a single species, right? Which is that where are each of these different species found? What is it that is driving these different species? Are there certain species found in one area, certain species found in another area? What is the thing that varies? Are there vulnerable species found in a certain area, small-bodied species found in another area? How do you do all of that? So recently, these set of models have taken off, which is what is called a bunch of models called the meta-community models, but essentially what is called the multi-species occupancy models, right? So multi-species occupancy models are joint species distribution models. Before this, I should, some of you asked questions about competition, etc. Multi-species occupancy models can also be two-species occupancy models, where you just take two species and like tiger and leopard, say, and say there's one species, competitor species, right? Does one species determine where the other species is going to be or is not going to be? Host and pathogen, yeah, exactly. Is there, like, how do these two species align? You have identified, say, a host, you've identified a pathogen, and how do they align with each other in space? So there are two species models, but as of now, we'll be talking about multi-species occupancy models, where you can go out, and this is an example of birds, which Varun had spoken about earlier. Byung Sem has done this for his PhD work. So essentially, there are about, I think, 128 species that he looked at. And you can take all of the species in a community, stack them one on top of each other, and look at all of them together as a community rather than separately and individually. So you simultaneously estimate occupancy and detection parameters. By now, everybody knows what occupancy and detection parameters are. You essentially combine, you sharing information across all of the species. This uses a Bayesian approach. I think there are likelihood approaches, maximum likelihood approaches as well, but then there are some really strong models that use a Bayesian approach to estimate the parameters. And you can get at the influence of covariates on the different species. So you can look at, does canopy cover influence all of these species or some of the species? You can look at, does bamboo influence the species? You can also look at, okay, where there is high canopy, is it the larger species that are being influenced by canopy? Or is it the species with, which are able to have more dispersal capability that's being influenced by these? So you can even look at things like that. All right, what's a meta-community? Very quickly, say you have 3 different patches like this. These are 3 different patches. You have some bird, it's only present in these 2. Different bird is only present over here. So these are your individual species. Like that, you have a number of different birds in your community, right? So you can include like some rare species, but there are also, this is your main issue, there are these undetected or missed species. This entire thing, the entire set of birds in one patch is your community. The entire set of birds in your entire area is your meta-community, right? So it includes this, and this. All of the set of birds in these areas is your meta-community. So the occupancy model, the multi-species occupancy model uses what is called data augmentation. That is, it says, there are a number of birds that I'm going to, which are undetected, unnamed birds that I'm just going to plug into this system and say these are not detected. And then it uses your data to code whether those added birds belong in your meta-community or they belong outside your meta-community. Just belong in your meta-community or not. The data that I use, let me just say this, the data that I use is the same thing as you used before. It's detection histories like you saw before, but stacked for all the species put together. So essentially what it has is just 3 levels. One is your entire set of species, right? Your species that you've detected, and this added bunch of species, and you have an indicator to see, is that species part of your meta-community or not? Then for each location, is the species which is inside your meta-community, is it present in location 1, location 2, location 3, location 4 or not? Once you have that, if species 1 is present in location 1, does it get detected by you when you go on occasion 1, occasion 2, occasion 3 and occasion 4 or not? That's it, that's all it does. So it takes a bunch of species, is it actually present in your meta-community? If it is there, is it present in location 1, location 2, location 3? If it is in location 1, do you detect it when you come in each of your times? From that, these are your 3 different things. From that, so your data, this YIJK is your detection history. That's for each of the different species. Your repeat visits, just as how repeat visits for occupancy for a single species allowed you to assess all these different things for a single species, same thing it allows you to assess your detection probability for multiple species. Once you've got that, you go to this Z value and this psi value, which is your probability of occupancy for each species in each location, and an indicator of whether it is present in each location or not. For each species, is it present in each location? So if you sum it up, across, if you sum it up for each location, it will give you the species richness. Yeah. If you sum it up for each location, it gives you your species richness. If you sum up for each species, it gives you the proportion of sites occupied, the total number of sites occupied. And same way as you could model it earlier, you can model it based on whatever covariates you decide, not whatever covariates you want, whatever covariates are biologically, ecologically plausible. Then you look at whether the species is there, the highest level, right? Whether the species is there in your meta community or not. The summation of that gives you the total number of species in your meta community across all of your different sites. So you may have heard, I just want to quickly, I'll come back to this, but you may have heard some of these terms. Alpha diversity, species richness is the total number of that, it's that summation of that Zi, total number of species in a site. Gamma diversity, meta community species richness, total number of species across all of your site. And your beta diversity is how that changes from site to site. Okay, so these MSOMs are really cool. They basically estimate the detection probabilities. You're not using any estimator, accumulation, nothing. You're directly estimating your detection probability. Can be modeled based on covariates. Your species distribution can be modeled based on covariates. You can get at species richness based on those, like modeling whether the species is present in a particular site or not. At the end of it, you get something that, you can get something that looks like this as well, which is species-specific site responses, like whether a particular species, how is it responding to a particular covariate. So if it is canopy cover, is it responding positively or negatively to canopy cover? This is for 128 species and for multiple covariates. You can also see certain types of species. So these are species, this is Hanwing index, which is how much can a species disperse. So species with low HWI have low dispersal ability, high HWI have high dispersal ability, and that essentially influences whether a species responds to, for instance, agricultural areas and protected areas or not. So it's the species which are not good dispersers are responding to whether there is a forest or not, and the species which are good dispersers are not responding as strongly. So you can look at things like that, right? And then just, I won't say too much about diversity indices. There are standard diversity indices which you can look up. There's a Simpson's index which is used for various different things. Shannon-Weiner index, Weiner index. These are all based on whether the animal is there and how many of the animal are there. And just to add that currently there are also multi-species abundance models, which I won't get into too much because I don't think they've been developed very, very greatly, but there are multi-species abundance models, which means that the same way like you plug a bunch of occupancy models together, you can also plug species abundance models together and incorporate these things, these types of error into your abundance models as well. That's it. You guys can go for coffee.
Just a quick question, Divya. So these SMOMs,
multi-species occupancy models.
Multi-species occupancy models can also be used to, for example, like in Biang's work, you can actually even think of species that are like shuttle species, maybe present across habitats, which could be the ones transmitting pathogens, or I mean, you could have hypotheses based on these results. You could actually propose these species could be the agents through which pathogens are spreading, because they're more generalist, present in more habitats, and things like that, which you could then test, say with phylogenies of the pathogen, which is a completely independent unsharing or something like that.
Yeah, I think you could even link the two. Maybe if you get like parasite load of the different species and then include that, asking the question, are the species which have a higher parasite load, are they found in like these, in multiple areas? So are they found in agricultural lands, or are they found near human habitation or not? And then there may be a subset of them which are, and those are your really, really important species that you're thinking of. And then there are a subset of species which have high parasite load, but they're not really found near where people are. So they may not be your species of interest. So you can look at those kind of things.
And just out of curiosity, has anyone also applied these models to vectors as well? So hosts, vectors, and pathogens.
I don't know. I don't know. Have they? Do you know?
Levels, right?
Yeah.
Or does it not matter?
No, if we are able to characterize the host, the vector, and the pathogen, through different whatever methods, then the model can easily be applied to all three. They're all part of the community effectively. Yeah. And like I was saying earlier, so you can look at what is the probability of one species being present given the other one being there, those kind of things, and also like a species interaction factor where, that quantifies that relationship between these. So host and vector, vector and pathogen, and so on.
There's been like a really cool long-term modeling that's been done also on these ephemeral wetlands. So that's habitat and the species that inhabit them, I think mallards, which is somewhat similar. So basically only if the habitat is there can the mallards be there. So it's a conditional thing, which I think is somewhat similar to here. Only if the host keeps moving around, it can be over here, it can be over there. And only if the host is there can the parasite be there. So it might be similar to that. But because as long as you have a very clear hierarchical model, and because these models have been developed in a somewhat simple way, but really very strong foundational way, and then there have been so many extensions to it, I think that as long as you have those initial conditionalities and probabilities really clear, I think that can be done.
Thanks so much. Thank you. So we'll take a brief break and be back in about 15 minutes. Thanks, Divya and Varun. That was
Thank you.
really
Thank you,
good. Yeah.
everybody.